Okay. So hello everyone and welcome for this month's new scientific news. Let me start by wishing everyone a happy new year, and I hope that 2025 is a very nice year for you, that you have a lot of health and a lot of happiness this year. And of course, I also hope for a lot of progress on aging this year, obviously. So the first thing is that I made a promise for myself this year that I will start a newsletter. So that was one of my, good promises for the year, and I have done it. I have published the first edition 2 days ago. So you can check that out. I send it to the mailing list, and it's on my LinkedIn, profile. So another thing is that Aubrey has published the last results of the RMR1 trial. So there are, I think, like 3 or whatever mice, 2-3 mice still alive. So it's pretty much every mice is dead, but they haven't done statistical analysis yet. So we still have to be careful because we don't have that data. But here you see the data for the males. And you can basically see in red, that's when you combine all the interventions together, the very thick red line. And the very thick blue line is when you give nothing to the mice. And you can see that, well, except for these very few individuals here at the extreme end, over the whole lifespan, all interventions do better than than nothing, sorry. So then the control. What you also see is that if you look at the orange line, there are so many curves, it's very hard to find them, but here you can follow it. So that's rapamycin alone, and rapamycin alone sits somewhere between the control and all interventions here. However, at the very end, you can see it's actually outliving everything. And another interesting thing is here, when you only do MTERT, you actually see that at the beginning there is not so much difference, but then the mice actually die younger. So that's a bit unexpected as well. So there are some things in this data that are difficult to explain. And when we look at the female data, so again, the same thing. The thick red curve is all interventions combined. The thick blue is nothing, is a control. And then you can clearly see again that everything combined is better than control. So that's, again, very good. When you look at rapamycin alone, the orange curve, we see it pretty much falls together with the red curve. So rapamycin alone works equally well as everything. And that's a bit, disappointing, maybe. You can also see, for example, here, MTERT alone in male mice, remember, it was decreasing lifespan, but in female mice, it's not. So there is a clear difference between female and male here. So there was this documentary, Don't Die, by Brian Johnson. I have watched it. There were some very nice, very short interviews in there with Andrea Maier, with Matt Caimberlain, with Joao Pedro, and with several other people. So I thought that was good and positive in the message. Overall, I would say I found the documentary positive. There is a little bit of cause of risk that some aspects, like at a certain point it's mentioned that it's like a little bit cultish, and that could be seen as negative. So there is a little bit but overall I will say I found it a relatively positive documentary.
Can I ask something about? Did you get the impression? I get the impression that there's a lot of interest in this person, but that also people might think that you have to be very wealthy to have any life extension benefits or whatever.
Yeah, that's another point that the documentary may kind of reinforce, and the reality, of course, is that you can achieve 90% or more of everything of benefit for basically nothing in money, and then you are chasing the tiny amount which costs a lot of money, because the 90% of all the health benefits and anti-aging benefits is simply, you know, exercising and having a healthy diet that is not necessarily very expensive, right? And then it's the tiny little things that you can add to that start to add up the cost. So, you know, that's indeed something that the public may not understand. Sadly, yesterday or today, I don't know exactly when, the oldest person on Earth, who was only 116 in Japan, died. And there is also going to be a journal club this month. It's going to be on the use of aging clocks, plasma protein-based aging clocks for organ-specific measurements of aging. On the 23rd of January at 5 to 6 p.m. In Central European time. And if you want to attend, you can send me a message. I will have a Zoom link for you. And there is also the upcoming Global Health Span Summit in Riyadh, Saudi Arabia, next month, in exactly a month from now. So the first news is a bit depressing news, because what it says is that among 183 countries, we actually see a widening gap between health span and lifespan. So indicating that the health of the population is decreasing due to a rise in non-communicable diseases. And I assume that is an assumption that, because it's not discussed in the abstract, that this is mainly due to lifestyle changes in the population. So that you see here, for example, the rise of obesity and associated diseases. This was a...
Maybe I want to make a precision about this, because what they say, yeah, the lifespan, the health span, lifespan gap increases, but it increases because the lifespan increases. So it's like for each 4 or 5 years, you have 3 or 4 years of good health. But it's kind of logical. Proportionally, it doesn't change so much. That's a remark. It's not like, okay, you live longer, but you live shorter life, healthier, healthy. You live a longer life, healthier, and an even much longer life, not healthy. Just to precise.
Yes, yes. But we possibly indeed also will see that you know, the rise of obesity, for example, in the United States, especially less in Europe, will have negative effects there on the healthy. Yes. So here, this is a paper from Kenneth Rockwood. He was the inventor of the frailty index. So the frailty index is a way in humans to clinically determine if someone is frail or not. And so they looked at a bunch of individuals, 47,000. And what they found is that around the age of 75, you see like a tipping point where suddenly the health really declines. So here in a mice paper, they looked at 1,576 male and female outbred Swiss mice that were subjected or not subjected to a high-fat, high-sucrose diet, so good diet or a bad diet. And then they looked at different biomarkers of aging, and they did that at multiple ages of 6 months, 12 months, 18 months, and 24 months. And what they found is that with aging, there was locomotion patterns and other multifunctional aging parameters that they could observe as aging biomarkers. In this paper, you know, there is epigenetic clocks, and the epigenetic clocks are basically all based on DNA methylation. But next to that, you also have modifications of the histones, the proteins around which the DNA is bound. And in this paper, they actually developed epigenetic clocks based on these histone marks rather than the methylation marks. And they found that these clocks were very accurate. And they also found that when you look at the genes, at the regions where these histones are modified, you can actually find that these genes are enriched for aging-related genes. And so that's interesting. Here they develop a single-cell RNA-seq aging clock to predict lung aging. And they find that for non-smokers, endothelial cells, but for smokers, epithelial cells for non-smokers and endothelial cells, so that's the cells that line the blood vessels, are best for predicting the age in smokers. Here they develop metabolomic age clocks. So they looked at humans, plasma samples with NMR. They looked at 168 metabolites from 225,000 middle-aged and older people with a mean age of 56 or 57 almost years. And they used those data to make 17 different algorithms, and the best algorithm was this mile age that was best able to predict the associations with health. So here in this paper, they looked at the NAD pools inside the cell, and so you have NAD inside the cytoplasm, so the liquid inside the cell, but you also have it in specific cell organelles, such as mitochondria. And what they did is they artificially depleted NAD in these various locations inside the cell. So the cytosol, the endoplasmatic reticulum, the peroxisomes, and so on. And they had decreases in NAD concentrations of over half. And they basically found that only if you decreased it in the mitochondria, then you develop problems. But in the other pools, it was contracted, it was buffered. So pretty much they say that the decline in NAD levels with age is probably well tolerated, except if the vulnerable mitochondrial pool is being affected. Here they look at a whole bunch of different interventions, including genetic interventions like the growth hormone knockout, knockout mice, the Snell dwarf mutant mice, the PPA knockout mice, but also pharmaceutical interventions like rapamycin, canegliflozin, meclizin, and acarbose. And also calorie restriction. And in all of these interventions, they found that there was a decrease in p65 levels. So p65 is part of of nuclear factor kappa B, NF kappa B here, and that's one of the main controllers of inflammation. So all of these things are decreasing inflammatory signaling. However, 17-alpha-estradiol and astaxanthin, two other interventions known to extend lifespan, did not have that effect. So the conclusion of this paper is that lots of interventions that actually extend lifespan are all, having the similarity of decreasing this NF-kappa B activity. So here they looked at whales, and there are of this species there are two subspecies: the southern and the northern one. And the southern one has a very long lifespan, and the northern one not so much. However, this difference is, according to them, probably mostly due to human activity and not necessarily due to an inherent biological difference. And so they say that hence the extreme longevity in whales may actually be underestimated because of human activity. So we are artificially, keeping the populations young by killing off whales. Here what they did is they developed mice, a knockout of this enzyme, which is needed for glutathione synthesis. And when you do that, you have 70 to 90% reduction in glutathione levels, which is quite extreme reduction in glutathione levels. And surprisingly enough, these mice actually do better. They are protected from age-related declines in motor function, for example. They have lower levels of inflammation going on and things like that, which is surprising because, for example, a lot of people have been supplementing with N-acetylcysteine to increase glutathione levels to counteract aging, and here you see the opposite. And this is probably because, again, when you decrease these levels of glutathione, you will cause a hormetic type of stress, probably on the body, a low level of oxidative stress, which will then probably activate cytoprotective mechanisms to increase the lifespan and the health. Yeah, this is not so interesting. Let's skip it. Here in this paper, they find that mitochondrial health and also chronic stress influence telomere attrition rates and telomerase activity. So if you have better mitochondrial health, you have longer telomeres. If you have less stress, you also have longer telomeres. Now there are two papers that I want to touch upon. Osteoarthritis. So osteoarthritis is when the cartilage in your joints gets worn down. So it's a type of wear and tear that happens. But despite it being wear and tear, it can be influenced by signaling processes. And it was already known that Wnt signaling is involved in this. And here but it wasn't understood how it exactly worked. And here they identified that Wnt signaling causes the production of IGF-1, insulin-like growth factor 1. And I mean, every one of you probably knows about this because this is one of the main things that is, signaling processes in aging. And so this IGF-1 gets locally produced by the synaptocytes, I think. So the cells there, or the chondrocytes. So here, the chondrocytes. So it gets produced there, and then it accelerates this osteoarthritis process. And here another paper where they find that this deficiency in, I assume it's an enzyme, I haven't looked it up, that causes mitochondrial dysfunction, and that then causes senescence of the synoviocytes, and that drives osteoarthritis progression. So role for cellular senescence in osteoarthritis. So here they look at sepsis. So sepsis is something that has a very high mortality rate. So that's, a lot of people end up in the ICU with sepsis. And they looked at these people and they classified them in high and low aging groups based on, gene expression that was happening in them. So I'm not familiar with this exact thing here that they did. It must be it's a previously published method. They classified them in these two groups, and they found that the high aging group were doing worse than the not so aging group. And they also found that thalidomide, which you might remember is this drug that caused birth defects when you gave it to pregnant mothers, that this drug actually significantly ameliorated lipopolysaccharides, so LPS, which is an inflammatory molecule that is found in the cell membrane of bacteria. So when you inject LPS, you get inflammation, and thalidomide significantly prevented that, and that prevented organ injury and cellular senescence in the lung and in the kidney of treated, I think, mice. That must be, because that's not going to be in humans. Here they find the role for cellular senescence in urinary tract dysfunction with age. Here, this is a paper from Oisin Biotechnologies, where they describe their gene therapy that can be used as a senolytic. So a lot of the so you have either small molecule senolytics, or else you have mice that are genetically engineered to have like a suicide gene in them that gets activated during senescence. But of course, that's not very applicable to humans as suicide gene, unless you can do it by gene therapy, and that's exactly what they did here. So they did a gene therapy with an inducible Cas9 suicide gene that was under the control of either P53 or P16, INK4A. So two promoters that get activated during senescence. And then they saw a significant decrease in the burden of senescent cells in mice that were treated. And this led to a 123% increase in post-treatment, be careful, post-treatment survival of the animals that were given this senolytic treatment. The mice also had significant reduced frailty, increased physical function, improved heart health, and a threefold reduction in tumor incidence. And here they find that mitochondrial metabolism is necessary to actually drive the SASP, the senescence-associated synchrotry phenotype, and that suppressing these changes in mitochondrial metabolism could actually prevent the SASP without killing off the senescent cells. So it's a way to diffuse the damage that senescent cells do. And here they find that metformin prevents actually chromatin fragments from leaving the nucleus of the cell when these chromatin fragments get into the cytoplasm. And so this is something that increases when you get older. When they get into the cytoplasm, they activate cGAS-STING, and that then drives senescence-associated inflammation. So when you give metformin, you prevent that, and so you reduce this inflammatory phenotype of senescent cells. So here they find that in senescent cells, a lot of DNA repair processes are completely suppressed. So they find that here the whole list is NER, PER, non-homologous end joining, homologous recombination repair, mismatch repair, and interstrand crosslink repair are all being decreased in senescent cells compared to non-senescent cells. And here a bit surprising, but Hydra actually has higher rates of mutation than mammals have. And this may actually have helped this Hydra in the lab to actually undergo a positive selection for being better able to live under laboratory conditions compared to the wild conditions. And then there are two back-to-back papers on lithocholic acid in Nature, where they demonstrate that this compound can act as a calorie restriction mimetic. So calorie restriction actually causes an increase in lithocholic acid. It's a metabolite of the bile salts. And so when you feed mice with this lithocholic acid, you get lifespan extension and health span extension. And this paper is focusing more on the mechanisms by which this happens. Here they looked at the short-lived turquoise killifish, and they find that amyloid beta precursor protein is actually contributing to brain aging in these fishes. Here they find the positive sorry, a negative, I should say, association between cardiorespiratory fitness and dementia risk. So if you are more fit, you have a lower risk for dementia. And here they find a positive association between the PM2.5, so that's the fine particulate matter, the fine dust, and frailty. So if you live in an area with a lot of fine dust pollution, you will have an accelerated frailty or a higher risk of becoming frail, I should say. And here, yeah, they find that poor sleep quality is also positively associated with the development of sarcopenia. C. elegans aging research, I'm only going to touch on one paper here, and that's this one. So when you take C. elegans, they are all genetic clones of each other. They are completely identical, and you grow them in exactly the same conditions. And yet, of course, you still see the normal pattern of aging, where some animals die young and others live longer. And what is really surprising is that you can actually, at a younger age, you can already predict which of the animals is going to live long and which is going to live short. And you can do that by looking at specific biomarkers. And here they looked at 4 biomarkers, this LIN4, this MIR243, this MIR240-786, and autofluorescence. So these 4 were used. 2 of them are positively and 2 are negatively correlated with lifespan. And when you do that, you can literally separate animals who are going to live short from those who are going to live long. Yes, so I'm going to stick it to that. Skipping all this. In the reviews, there is, I would like to point out a few things. Here is an expert consensus statement on biomarkers that might be interesting. Here is one for considerations on combination therapy in Alzheimer's clinical trials. And here is one that questions at what age longevity treatments will actually still work. And then, yeah, you can take a look at the other reviews. Here is a paper from Alex Savaronkov on the progress pitfalls and the impact of AI on clinical trials. Here is a paper showing that fasting mimicking diets are beneficial against breast cancer. And here is a paper that shows that if you remove telomerase, that actually tumors are not proliferating anymore. So actually, you know, telomerase is involved in driving melanoma cancer in the zebrafish model. And that's it for this month. So are there questions?
Thank you, Sven. I have, it's not a question, it's more a comment and one suggestion. The comment is, I didn't know about the death of the oldest woman. I took a look at the surviving oldest people. It's really depressing. Hein? So from the 100 oldest women ever, there are only 4 alive now, and the oldest of the 4 is only the 21st of all ever. And from the 100 oldest men, the first still alive is the 57th, and there are only 2 men in the 100 oldest men. So when you know that I say since many years that sadly we don't progress for maximal lifespan, but it's even more true today than a few years ago. This is probably the most depressing aspect concerning longevity that you can notice. So many progress, so many scientific articles, so many conferences, and no progress for longevity, maximal lifespan at least. And then my suggestion was maybe for the next meetings you can do more slides with the big upcoming conferences. But yeah, now that I'm saying it's also not such an important idea. It's only for the upcoming in the close future. And are you going to try to go to Saudi Arabia? I'm going to try, or if it is possible for Nino, I'm going to propose to Nino to go. but I'm hesitating.
No, I'm not going to go. Victor is going to go. And I'm going to try this year to go to the Berlin conference and probably also to Copenhagen to ARDD.
Yeah, IRDD is
That's-
probably the most interesting. Yeah.
I had no idea that there was one in Saudi Arabia as well.
Yeah, it's close already. It's in less than 1 month. It's done in 1 month, precisely 3 and 4 February or something like that. Maybe
I will Google it.
4 and 5.
Yeah, 4 and 5. Yeah.
Do they have any like a fee to participate in Saudi Arabia?
What did you say?
Do they have a conference fee you have to pay to go there?
No, I didn't check.
I think you might need an invitation to go. I think there might not be a fee, but I think there is an invitation.
I was taking a look, and you have to ask to go.
Yeah. I checked the website, and indeed you have to.
Yeah, you have to ask, but I don't know. Nino or Sven or Shivanu, I don't know. Somebody told me there was a conference 1 or 2 years ago, and it looks to really empty. So it was strange. Okay.
Sven, did I understand you correctly that you plan to go to Berlin to the conference? Because I thought the conference in Berlin doesn't take place anymore.
Oh, okay. I didn't know. I assumed that will continue every year.
No, because Michael Grief, he somehow, maybe it's only rumors, but somehow he quit, I thought, like a couple of months ago. And I think he doesn't organize the conference anymore.
Oh, yeah.
But I'm not 100% sure.
He quit something?
Yeah, I think he closed down some of his investments and so on.
Oh.
Maybe.
But I'm not sure. I mean, I don't have the insights. I just read it in the Vitalist chat, and Aubrey wrote a sentence about it. And yeah.
Anyway, what is sure, I didn't see any announcement for Berlin the last months. That's for sure. Yeah. Okay. Yes, there is a. I will put the link on the on the chat here. There is a Facebook group for conferences. For conferences on longevity, of course. I mean, I will try to find it again and put. Sorry, so I was speaking a lot. Other questions, remarks, and so on.
Yeah, just a comment regarding the presentation of Sven. The Netflix documentary, the subtitle is "The Man Who Wants to Live Forever." So I suppose there are billions of people who want to live forever. So the way they put this subtitle gives the wrong impression that it's something very particular, from Brian Johnson, the man who wants to live forever. So it's a wrong message. I mean, it says a lot about the problems in the mass media about with the rejuvenation or longevity movement. So I just wanted to point out this, because it's a bit ridiculous, the men who want to live forever, when billions of people would like to live forever. I suppose billions of people don't want to live forever also, but it's not a particular thing of Brian Johnson.
Mm-hmm.
Yeah.
I think one of the things is he wants to live forever in the spotlight.
Yeah. Okay.
Well, he acts on it. I mean, maybe that's the reason. Like, one thing is wishful thinking, and one is actually acting on what you want to do.
He has the money.
And he has the strategy. So, and he has the money. Is that what Livia says?
No, obviously, he has the money.
Okay. Yeah. But the thing is that he acts on what he wants.
Yeah. Actually, with what he is doing, if there is no medical progress, he is of course not going to live forever anyway. That's for sure. But that's the same problem for everybody. And like Sven said, actually, the most things that you can do at the moment, you don't need a lot of money for this. And the one that you cannot do without a lot of money, they are quite risky in the most cases.
Yeah, and he's spending, of course, most of that money on actually all the the measurements he does. So the fact, I mean, he has like this whole clinic in his home with echography machines and you name it, right? A centrifuge, when they take his blood, they centrifuge it at his home and you name it. And of course, that's also all not necessary, right? I mean, so that adds up all this cost, and then all the doctors he has, and the stuff, and you name it. So there is a lot of money he spends that is not really going to be that significant on any health benefits.
And there is also another aspect. Somebody doing so much efforts, for most people it is impossible, and I would not be surprised that one day he will also stop, because like 5 or 10 hours exercise, and we will see anyway.
Well, there's another thing too, which is that first of all, he's talking about living forever is just plain philosophically ridiculous. It makes the anti-aging, the longevity movement look ridiculous.
But I think also that's just they want to have a title, like they will speak about immortality, even if the documentary is only about longevity, healthy lifespan, and so on. Okay. Anyway, other comments?
Well, it's like you said, we're actually going backwards in terms of the oldest person on Earth. and we're talking about living forever. Why don't we hit 130 first?
Yeah, for sure.
In good health, mind you. Those old people that are living, they're not exactly running marathons.
Okido.
By the way, when you mentioned marathons, maybe one thing I could have put in the scientific news, but I forgot, was there is a woman in Belgium, and she ran a marathon every single day for the whole year. And she was not super young. I mean, she was not old, but she was middle-aged.
She's like 55, I think, or something like that.
Something, in her 50s.
But I mean, it's like how are her joints after running marathon every day?
Sorry?
No, but if she runs it every day, I mean, it's like destroying your joints.
Yeah, she did it for breast cancer, I think. I help funding breast cancer. It's definitely not recommended for health.
Well, typically, like as a person who actually ran marathon in the past, typically you need several days to recover for it. And if she does it every day, I don't know, it's really damaging because she needs recovery.
I put the information online. I don't know why he is Al Jazeera, because it was the first one in my chat, but it's a Belgian woman. It's a Belgian woman, so it's great. A bit of chauvinism, I'm joking, of course. Other comments concerning the scientific news?